In this video, we're going to continue on with Chapter 4, which is talking about experiments, studies, and how to sample. Okay, it's sort of the second step in the statistical process that we go through in this class. So now we're going to talk about good and bad sampling. Okay, so when we do sampling, when we are taking data from a population, because of course we can't sample the whole population, we have to sample part of it. There are good and bad ways to sample. So let's talk about the bad ways to sample first. We spent our last time talking about good ways to sample. Now we'll talk about bad ways. So bad sampling happens when there's some sort of bias that's involved in the way that we sample. Okay? And bias happens a lot when you're sampling. You have to be very careful about how you sample and sort of how you're going about taking data. Because if you get bad data through biased samples, then the results that you are going to get are also biased. Okay, and that's not something that most people want. So there are a few types of bias that can happen here, and this is certainly not an exhaustive list, but it's some of the main ones. So there's sampling bias, okay, when non-random sampling or under coverage happens. So what does this mean? When you sample, sampling bias, non-random samples or non-random sampling happens when there's no sort of randomness involved in how you sample people. So we'll talk about this in just a second, but you can take what's called a convenience sample. Maybe you go out and then the first 10 people that you see, you ask the question to. There's no randomness involved there, and so that's not great. Or you could just pick your friends. Okay, that's probably even worse than the first 10 people because there is some randomness, I guess, there. But picking your friends, your family, when you're sampling is not a great idea. And that non randomness leads to undercoverage. And what that means is when your sample does not cover the whole population. So if we were talking about the city of New York again, and I only sample people from Queens, okay, one borough of New York, that leaves the rest of the boroughs out. And so then I'm not taking into account people from all over New York, just maybe the borough that I live in or something like that. Okay, so there's sampling bias, and then there's also non-response bias. Non-response bias is what you see happen a lot in something like class observations. Okay, when they ask you to answer questions about the class, how you thought it went, when you evaluate me. Okay, and so non-response bias is when some subjects won't participate in the study. Okay, and so what you get when you have non-response bias are the only people that respond to surveys. Usually, in general, non-response bias happens a lot. Okay, and when you only get people who respond to surveys because they had a really good experience or a really bad experience, not one in the middle, what you get is a biased response because not everyone participated. You may have gone to great lengths to pick out a random sample of people, but if only people who think a certain way will respond, you get really biased results. An example of this is the 2016 election, when almost every poll out there predicted that Hillary Clinton was going to win the election. What happened is they seemingly had major non response bias. People who had maybe decided to vote for Donald Trump but did not want to admit it for some reason or another to the poll people just didn't participate in their survey. And so the data that the pollsters got overwhelmingly favored Hillary Clinton when in reality it was not that way. Okay, so there's non-response bias. And then the third is response bias. And you might think that that's sort of opposite of non-response bias, but it actually talks about a different kind of bias. Response bias is when you ask questions or phrase questions in a certain way to make the subjects respond. Okay, so as an example, if you ask a question like, if you ask a question on a survey, like and the survey is maybe about abortion, okay, and if you ask the question like one biased question would be maybe, do you support women's right to choose whether or not they have the freedom to do something with their body or something like that? That is, of course, a biased question, or could be viewed as a biased question. Okay, and on the other side, you could ask a question like, do you support doctors ripping out babies out of the wombs in the 9th month, or something like that? A very biased question that's going to elicit a certain response. Especially the last question is a very like a visceral question, but both questions to some extent offer or look for a biased response. Okay, so that's response bias. When you can't perform an experiment, you take a survey, collect data that's already taken, okay, and so you have to watch out for these types of biases, especially there. Okay, so what are some techniques that lead to bad sampling? So one I talked about before was a convenience sample, okay, sampling the most convenient subjects. So that could be your friends, your family, the first 10 people that you see when you walk out of the building when you're trying to take a sample. Okay, so if I walked out of Dauphin and I asked the first 10 people there, that might not be so terrible because Dauphin is the humanities building on Shippensburg's campus, and most people have to take a humanities class. So there's a decent chance that those 10 people are fairly representative of the population if I was interested in Shippensburg. Whereas if I went to somewhere like Higas Fieldhouse, okay, and I had the first 10 people that walk out of there, most likely if you're in Higas Fieldhouse, you're probably you're probably involved in sports in some way or another. When you're in like a basketball or volleyball or wrestling or swimming or something that is takes place at Higas Fieldhouse. So when I get that sample, that's probably going to be biased towards athletes or in some way or another, and athletes may not be or hold the typical viewpoint of the whole campus. Okay, so convenient sampling, sampling the most convenient subject, is not generally a good idea. Volunteer sampling is when a sample is a sample where subjects volunteer to be in this sampling. So this is something like a Facebook poll or a Twitter poll or an Instagram poll, a social media poll, okay, or just something that you put on the internet and ask people to respond to. Okay, the only people who are going to respond are volunteers. They aren't. You aren't going up to them and asking them, to answer this question. They're volunteering to be in the sample. And so that is going to lead to biased results because the only people who are going to answer the survey are those who are interested in the subject being discussed. Okay? And so they will probably have a certain response to those questions that other people might not have. And so you're undersampling those people who may not be so interested in answering your questions. Okay, so those are the bad ways to experiment. Okay, convenient sampling and volunteer sampling. There are good ways to experiment as well. Okay, so bad sampling techniques. There are good ways, we talked about them: random sampling, cluster random sampling, stratified, okay, and then there are also bad ways: convenience and volunteer. Now, there are also good and bad ways to experiment, and we'll go over these fairly quickly here. So when you create an experiment, you often or you always need a control comparison group. And a control comparison group is a group of subjects in the experiment that take either the standard of care or a placebo drug. Okay? You need something to control or to compare to your treatment group. Okay, so a control group is important because you need to know how subjects would react under what are called normal operating conditions. So, how the body is going to react during this time, or how whatever will react. Especially, you need this control comparison group, especially because of something called the placebo effect. Okay? And the placebo effect occurs when subjects respond better to a placebo than they would to nothing at all. Okay? So you can often hear of stories where if you give a person some like colored water and tell them that it's beer, okay, if they don't know any better, they could drink it and then think they are drunk. Okay? Even though they didn't drink any alcohol, you can trick people into doing that. If you're taking a drug and you think that that's going to help, your body's going to respond to that, okay? And thus, you'll get a better outcome than you would taking nothing at all. So that's a placebo effect. And so that's why a control group is so important, because it may not be the case that nothing at all happens, but we want to make sure that the treatment group is actually getting better and it doesn't just have a placebo effect going on there as well. And so the next thing that's important, other than a control comparison group, is the idea of randomization. Okay? Randomly assigning subjects to the treatment and control groups is extremely important. And it's important because doing that often cancels out or mitigates the effect of, so it doesn't cancel out necessarily, but it mitigates the effect of different variables that we don't control or understand in our experiment. Okay? So it mitigates any bias from the experimenter, and it also mitigates different conditions that people may have. Okay? So you don't want to necessarily put all people who are diabetic in one group, okay, and all people who aren't diabetic in another group, because diabetes may have an effect on whatever you're testing. Okay, can balance groups on looking variables, and variables know that affect the response. Okay, so randomization is extremely important because it's the only way to make some causal inference on the response, and we'll talk about that in just a second. Some more good ways to experiment, or terms that are involved in a good experiment, is blinding. So you may have heard of a blinded study, or even better, a double-blinded study. Blinding happens when the patients don't know which group they're assigned. Okay? Because, of course, if you know you're assigned to the placebo group, then that placebo effect kind of goes away, okay? And it changes the experiment in some way. So it mitigates any psychological effects that you might have of knowing that you're in a bad group or a good group. Okay? And then, of course, double blinding is sort of optimal when neither the subject nor the people having contact with the subject know which group the subject belongs to. So if you're in a human trial and you have nurses that are helping these patients doing whatever, if those nurses also don't know what's happening, that again mitigates any sort of side effect that could be passed on from the nurse to the person. If the nurse knows that they're in a control group, in a placebo group, then they might treat that person differently than if they're in the actual treatment group. Okay, so double blinding is optimal. Okay, so if I go back here to randomization, if everything is done correctly, then you are allowed to, or you may be able to, make a causal relationship between the predictors and the response, or predictor and response, that you're interested in or that you're studying. Okay, so if you could, for instance, look at smoking and lung cancer, and you could say that you balanced out all the groups, all lurking variables, and you're sure that nothing else could cause lung cancer other than if you smoke or not, then you can make a causal relationship, okay, or make a causal claim, being that smoking causes lung cancer, not just that the two are associated with each other, like we explored in the last chapter. Okay, so it's very hard to make those causal relationships, and to do so, you need to use an experiment type thing so that you can control for all the other different lurking variables. Okay, and so at the end here, I put a summary from the book that you can look at, and talks about the key parts of a good experiment that I talked about in these last few slides. Okay, that's the end of chapter 4, okay, and really the end of a section of the book. That first section is talking about describing data. In the next section, we're going to get to start talking about modeling data, whatever that means.